So now, after understanding the process of oogenesis, let us see what all things are present in the egg. So this is the structure of human egg. And because we are seeing the inner structure also, you can say it is a section or a vertical section. The egg is spherical. It has its plasma membrane, and during growth phase there is one more membrane which is secreted around it, and this membrane is almost fused with the plasma membrane, almost fused. So they are like very close to each other, and that is why we do not want to show any gap. So this pink membrane which we have drawn here is vitelline membrane. The inner part is filled with cytoplasmic material which is known as ooplasm. This ooplasm is divided into two parts. The outer part is dense and granular. There are granules here. This is known as the egg cortex or exoplasm. And the inner one is smoother without granules rather, and is known as ooplasm or endoplasm. The nucleus becomes bilobed. This bilobed nucleus formation also takes place during growth phase. So nucleus, it becomes large and bilobed. In case of sperm, the nucleus becomes compact or condensed by losing nucleoplasm. In case of egg, the nucleus becomes bigger by taking or making more and more nucleoplasm, and this nucleus is a bilobed nucleus which is known as germinal vesicle. And as you can see here, we have not drawn the nucleus in the center. It is slightly towards one side. It is eccentric. This eccentric position gives the egg a polarity. So the side which is towards the nucleus, that means this side, is known as the animal pole, and the opposite side is known as the vegetal pole. This is due to the position of nucleus. Now why this positioning is important? Because the entry of sperm always takes place from the animal pole, and when the sperm enters, if it finds the nucleus right there, because ultimately nuclei have to fuse for fertilization first. So this is the place from where the entry of sperm will take place. Outside the vitelline membrane, there is one more membrane. It is non-cellular. This membrane is secreted by the follicular cells which are around this. It is known as zona pellucida. It is secreted by the follicular cells, and there is a space between vitelline membrane and this zona pellucida. It is known as perivitelline space. if you remember, we were talking about the structure of ovary. We said in the ovary there are clusters of follicles. One follicle is going to enlarge and all other follicles keep surrounding it. This bigger cell is going to form the secondary oocyte and then the egg. So, these follicles which are surrounding it are here also. So, in this case also there are follicles. So, these follicular cells, they are found around the egg. They are outside zona pellucida. The arrangement of these follicular cells is such that they appear as if the cells are radiating, like you could see here, the inner layer is uniform and there are some cells which are sort of radiating out in the form of rays. So, these cells, follicular cells, multiple layers, and they seem to be radiating from the egg. This layer is known as corona radiata. This is the layer of follicles. It is made up of cells, so it is a cellular layer. So, in the egg there is cytoplasm, which is divided into two zones. The outer zone is denser, granular, it is known as cortex or exoplasm. The inner is more or less smooth, non-granular, it is known as endoplasm or ooplasm. There is a big bilobed nucleus which is eccentric, and this eccentric position gives the egg a polarity. The side towards the nucleus is known as animal pole and the opposite side is vegetal pole. Sperm enters from the animal pole. Outside the plasma membrane there is a vitelline membrane which is secreted during the growth phase. Outside vitelline membrane there is one more layer which is called zona pellucida. It is non-cellular. That means it is not made up of cells and it is secreted by the follicles. Between vitelline membrane and zona pellucida there is a gap or space which is known as perivitelline space, and outside zona pellucida there are many follicular cells. They make one layer and the cells of this layer appear to be radiating outwards from the egg. This layer, which is cellular, made up of follicles, is known as corona radiata. So, this is the structure of the egg, or we can say the secondary oocyte. When we draw secondary oocyte, that means what has happened? Meiosis 1 has taken place. If you recall oogenesis, after growth it was primary oocyte which is diploid. Then when it undergoes meiosis 1, first polar body and the secondary oocyte is formed. So, if this secondary oocyte is there, everything is happening in this cluster of the cells. So, suppose this secondary oocyte is, or let us take this as the primary oocyte. Suppose we say this is primary oocyte, and this primary oocyte undergoes meiosis 1. So, it will give rise to the secondary oocyte and a polar body, and this, let me use another color here for secondary oocyte, and this tiny structure which is polar body, and this entire thing is surrounded by follicles. So, in this also, outside the vitelline membrane we can see one polar body. So, this one thing which we have to remember, if we are drawing a secondary oocyte, we have to remember after which stage secondary oocyte is formed. It is formed after meiosis 1. That means secondary oocyte is formed and a polar body is also formed. So we can, or we should draw this polar body here. If this is secondary oocyte, this polar body should be labeled as the first polar body. And if we are drawing an egg, then also one polar body is formed, so then it should be labeled as second polar body. What has happened to the first one? The first one by that time degenerates. So we have to remember when we are labeling it, if it is secondary oocyte, then the first polar body will be, or the polar body will be labeled as first. If this is egg, then the polar body should be labeled as the second polar body. So this is the structure of the human egg. Now let us also talk about the types of eggs. Types not in the sense of eggs containing or some other thing containing. All eggs are eggs containing eggs. They differ only in their yolk content or distribution of yolk. So if we talk about the types of eggs, and the basis on which we are seeing the types is on the basis of yolk content. If the yolk content is very less, negligible or absent, then such eggs are known as microlecithal or alecithal. It's almost negligible, as good as no yolk. Such eggs are known as microlecithal or alecithal. Human eggs are microlecithal. So we find this in humans. If the yolk quantity is moderate, then they are known as mesolecithal. Now this will, instead of trying to mug up the examples of all these types of eggs, if egg has no yolk or negligible yolk, what is the alternative to provide nourishment to the developing embryo? Obviously, it is that the female or the mother is going to supply that nourishment throughout the period. So in eutherians, like placental animals like human beings, because there is a placenta which connects the fetus to the mother's body, there is a regular supply of nourishment. So even if the egg is without any yolk, it is not going to affect the fetal development. So examples here would be all placental animals like human beings. Mesolecithal means the egg has less yolk, moderate yolk, not sufficient for the complete development. Do we know of some animals where the egg, or maybe not the example, but some animals where the egg has insufficient quantity of yolk, so in the egg partial development takes place and an intermediate stage hatches from the egg. Then this stage collects more nourishment, undergoes one more change of development, and then adult is formed. What happens in case of insects? They lay eggs, there is little yolk in it. That yolk is sufficient only for development till larval stage. So what comes out of the egg is larva. Then what do these larvae do? The larvae keep on eating. They are voracious eaters. They eat and collect that nourishment. Why are they collecting it? They did not have that much of additional yolk for their development. That's why it is an intermediate stage. The larvae, they feed, collect nourishment, and then they change into another resting stage, resting from outside, which is the pupal stage. And now using that food which the larva has taken, further development will take place. So which type of organisms will produce mesolecithal eggs? The animals in which we see intermediate stages. Can we also call marsupials under this, like kangaroo? the eggs which are produced inside the body, they are with moderate to less yolk content, and that is why the fetus which comes out of the egg is incompletely developed. So, organisms in which a larval stage or an intermediate stage is seen, those eggs are mesolecithal. If the eggs have enough of yolk content, then they are known as megalecithal or polylecithal. The eggs of reptiles and birds, they contain significant quantity of yolk, and that is why from the egg what hatches is a small animal. It is smaller in size, but all the structures are formed. So, in birds and reptiles, here we can write the example of insects. So, megalecithal or polylecithal, such eggs are self-sufficient eggs, and they are also known as cladoic eggs. The eggs which are self-sufficient in the sense of the reserve food material, they are called cladoic egg. So, three types of eggs on the basis of the yolk content. If it is negligible or absent, microlecithal or alecithal. If it is moderate or less, then it is mesolecithal, and if it is in plenty, then megalecithal or polylecithal. This is on the basis of yolk content. Now, we can classify the eggs on the basis of the distribution of yolk. So, types of eggs on the basis of yolk distribution. If the yolk content is uniformly distributed, then such eggs are known as homolecithal. Where there is uniform distribution of yolk. Normally, microlecithal eggs are homolecithal because the yolk content is so less that it gets uniformly scattered in the ooplasm. Second type, if the yolk is towards one side. See, normally why this is important is the division, when this zygote will divide, when this egg gets fertilized and the zygote divides, it is the cytoplasm which has to divide, yolk does not divide. So, only cytoplasm, nucleus and cytoplasm. So, if the yolk is like plenty and everywhere, then the cytoplasmic division will get disturbed or it will not be proper, and that is why the yolk has to be either in one side or in the center, or if it is almost negligible, then uniform. So, that also affects the division, cytoplasmic division. That is why this distribution is also very, very important. If you have seen hen's egg, suppose we boil an egg and cut it vertically, the egg shape narrower from the upper end, wider from the bottom, and normally the yolk is drawn here. If you see the boiled egg vertically cut, you will find the yolk is on the lower side. That means it is towards one end. Such an egg which has yolk towards one side is known as telolecithal. The eggs of birds and reptiles are like this. In some animals, it could be like in the form of a disc, may not be like a circular structure, can be in the form of a disc. The third type, if it is in the center, so if this is the egg and the yolk is in the center, then it is known as centrolecithal. Centrolecithal eggs are found in insects. So homolecithal, uniform distribution. Almost all microlecithal eggs are homolecithal. Then telolecithal, normally they are polylecithal, yolk content is more. In case of birds and reptiles, the yolk content is towards one end. And centrolecithal eggs are normally mesolecithal, the yolk content is not that much and the yolk is in the center. So these are two classifications or types of eggs on the basis of one, yolk content, how much of it is there, and second, how is that yolk distributed. So three types. Now let us also talk about the egg membranes and see what all these membranes known as. Egg membranes or egg envelopes, the layers which are around the egg. They are divided or classified as primary, secondary and tertiary. These are egg membranes. The primary membrane, secondary membrane and tertiary membrane. Primary membranes are the ones which are secreted by the egg. Secreted by the egg. So which membrane should be considered as primary? We don't count plasma membrane because that is the basic membrane of any cell. So outside plasma membrane, vitelline membrane which is secreted during growth phase of oogenesis, that is secreted by the egg. So example of primary membrane is vitelline membrane. Secondary membranes are the ones which are secreted by the follicles or made up of follicles. So they are secreted by the follicles or made up of follicles. Around the vitelline membrane, there is one layer which is known as zona pellucida, non-cellular layer secreted by follicles. So example of this, zona pellucida, and the layer which is made up of follicles, that is corona radiata. And tertiary membranes are the ones which are secreted by the oviduct or fallopian tube. With the help of some glands. So there are glands which are present in the fallopian tube and the layer which they secrete are known as tertiary layers. In case of the eggs which we see in hens or reptiles, the eggs have shells around it. So the shell, or sometimes there is a mucilaginous cover around the egg, that is also a tertiary layer. So three types or three categories of layers around the egg: primary membrane, secondary membrane and tertiary membranes, depending upon which structure produces it. If it is by the egg, then primary; if it is by follicle, then secondary; and if it is structure other than this, that is part of fallopian tube, maybe a gland which is present in fallopian tube, then those membranes are known as tertiary membranes. So we have seen the types of the eggs on the basis of yolk content and their distribution, and also the membranes which are around it. Now let us talk about few more things which are a comparative thing between the egg and the sperm. So if we compare the eggs and sperms, or rather egg formation and sperm formation processes, the egg formation is by oogenesis So eggs are produced by oogenesis, in which one egg mother cell gives rise to one egg. So one oogonium produces one egg. In case of sperm formation, the process is called spermatogenesis, and one spermatogonium, that is sperm mother cell, gives rise to 4 sperms. Eggs are all identical. That means all eggs containing. All eggs are identical. Identical means eggs containing. Whereas sperms are of 2 types. One, eggs containing, that is gymnosperms, and Y containing, eggs containing gymnosperms, and Y containing androsperms. Eggs are non-motile, they do not move on their own. In the fallopian tube, they are conducted by the cilia which are present in the fallopian tube. So eggs are non-motile, whereas the sperms are motile, they have to swim up to the egg. So they are motile structures, they move. Now one more thing which we will talk about here as a comparative thing is the lifespan and viability. Lifespan means for how much duration will the egg survive, that is its life. Same with the sperm. And viability means for how much duration would it retain the power of getting fertilized or fertilizing. So lifespan in case of eggs is about 2 to 3 days. In case of sperms also, it is approximately same, about 2 days, 2 to 3 days. Viability, that means the duration for which they will attain or retain the power of fertilization. So viable, the eggs are viable for about 48 hours and the sperms are viable for about 72 hours. So the lifespan is more and viability is always less. So if they get fertilized within this time, it's okay. Otherwise they will remain alive, but they will lose the power of getting fertilized or fertilization. Structure of egg and sperm we have seen in detail. We have also seen the comparison between oogenesis and spermatogenesis. Oogenesis is a discontinuous process and spermatogenesis is a continuous process. This process gets completed in 74 days, whereas it starts in the fetal stage and it will get completed only if fertilization takes place. So these are some important things, and this is the first step of the sexual reproduction, that is gametogenesis. So we have just seen gametogenesis, and the next stage is once the gametes are formed, they will be fertilized. But before that, we will talk about menstrual cycle, which is seen in primate females, and estrous cycle, which is seen in non-primate mammals.